Hello, my name is Jason Adrians. Today is 2/28/2025, and this is Anatomy and Physiology of the Retina. The retina is truly a fascinating feature of the body. It often gets compared to a camera, but a camera just detects the light. The retina not only detects the light, but it also converts it into chemical messengers so that the brain can understand it. But before I go into the deeper physiological role of the retina, you kind of got to understand the entire anatomy of the eye in order to understand why the retina functions the way it does. The retina is this thin layer of tissue at the back of the eye that detects the light. Light goes through the eye, it goes through the cornea. The cornea's job is to focus the light so that it hits the back of the retina and somewhere called the fovea. And the light goes through the cornea, and then it goes through the pupil. The pupil size is determined by the iris, and the iris is the colored part of your eye. And then it goes through a feature called the lens. The lens further focuses the light towards the back part of the fovea, or towards the back part of the retina, called the fovea. The reason that the anatomy is structured the way it is to focus the light on the fovea and the macula in general is because the macula and fovea give us our sharpest vision. They have things called cone cells, and cone cells give us our sharpest vision. Light doesn't only hit the fovea, it does hit the peripheral retina, but the peripheral retina is what your peripheral vision is. With your peripheral vision, you can only detect motion and vague features of shapes and things in low light. You can't really read with your peripheral vision. You can read something that's directly in front of you, but something directly in front of you is that light is getting directed onto your fovea, which is why you can read something directly in front of you. The role of retina in vision. The role of the retina is to take the light signal that comes in from our eyes and convert it into something that the brain can understand. As you can see on the diagram on the left, the retina this is just the retina, and it has multiple cells each doing some different job. And I'm going to go cell by cell and explain what each one of these cells does. So I'm going to start with the pigment epithelium. The pigment epithelium basically just prevents scattering, and it recycles the pigments that excess light gets through. Excess light goes through the rods and cones, and then it recycles those pigments. The next cell in this diagram is the rods and cones. These are the photoreceptors. The photoreceptors are what start the signal process and basically create something that the brain can understand. So light comes in from the left, it goes through the ganglion cells and the bipolar cells, and it reaches the photoreceptors, the rods and cones. The rods, like this cell here, the rods are responsible for low light vision and just detecting motion and like having basic night vision. It can detect shapes. It's not very clear vision that this rod cell detects. And then cones work best in bright light, and they provide sharp, detailed vision. From there, the signal goes through, the signal gets generated in the photoreceptor cells, and then it goes to the bipolar cells. However, before it goes to the bipolar cells, these horizontal cells have a job of creating contrast in the signals. The best way that I can put this is the difference in the light comes in and it hits each and every one of these rod cells. Each and every one of these rod cells. So how the brain can tell the difference between what light hits this rod cell and this rod cell is that's the horizontal cell's job. It's to create contrast in the signals and then pass that signal on to the bipolar cells. So then the signal goes to the bipolar cells, and the bipolar cells basically pass it on to the ganglion cell. It just converges that signal and makes that signal more processable, I guess, by the brain. It simplifies it down. The amacrine cell has a very similar job to the horizontal cell in the role of it provides contrast, but it doesn't do the exact same thing because it's not from the photoreceptors to the bipolar cells, but it's from the bipolar cells to the ganglion cells. So then these ganglion cells have axons, and these axons converge at the optic disc, And that's what forms the optic nerve. And then the optic nerve then takes that signal and passes it back to the thalamus, and then thalamus takes that to the visual cortex, and the visual cortex is ultimately what processes the vision and creates an image. So our photoreceptors are a very important structure in the retina, and they have two main types, rods and cones. I briefly mentioned this in some of the other slides. Rods are mainly found in the peripheral retina, and they allow us to see in dim conditions. They are extremely sensitive to light, and they only allow us to see different shades of gray. Cones, on the other hand, allow us to see they require bright light, but they allow us to see detailed vision, or they give us detailed vision, and they allow us to see colors. There's three types of cones. There's red, green, and blue, and these three types of cones allow us to see the entire color spectrum that humans can see. A fun fact about this is colorblind people actually lack one of these types of cones, and then they can't see that specific color, or that spectrum of colors that lacks that cone gives other humans that have that cone. Cones are densely packed in the fovea, and basically that's what explains why we struggle to see in low light, but in bright light we have detailed vision. So now that we understand photoreceptors, the next step in the process is phototransduction. And phototransduction is ultimately how the signal goes from the photoreceptors to the brain. Unlike most neurons, photoreceptors are actually active in the dark, and then when light strikes them, then they alter their state, and that's what triggers the signal. So they actually are constantly releasing a neurotransmitter called glutamate, which turns off the signal, and then when light strikes them, it alters the glutamate, which then the bipolar cells, or it alters the glutamate release onto the bipolar cells, and the bipolar cells recognize this and start the signal. Or they signal the ganglion cells. But there's two different types of bipolar cells. There's on cells, which work in light, and off cells, which work in darkness. And that is the reason that we can see edges and patterns, and this is what allows us to interpret shapes and different things, and allows us to read, allows us to see things coming at us. The light is this altered response is what allows the signal to actually fire and allows the brain to interpret it. So then the signal goes from the bipolar cells to the ganglion cells, and then the ganglion cells are actually the first cell in this process that generate a true action potential. These other cells just alter the signals, and then the ganglion cells actually generate an action potential and send it down their axon. And the axon meets at the optic disc, and then they converge at the optic disc, and then the axons of these ganglion cells actually form the optic nerve, which then sends that signal to the brain and to the visual cortex, or actually to the brain, to the thalamus first, then to the visual cortex, and that's what allows the brain to process this signal. So why does this matter? The retina filters and refines visual information before it leaves the eye. As I mentioned in the first slide, a lot of people like to compare the retina to a camera, but actually it's a very organized structure, and this is what the light actually gets processed in the retina before it goes to the brain, because the brain can't just process pure light. So it's a very organized structure, and it has a lot of physiology just in this one little compartment of the eye. So this is a study aid that I created in Canva, and this basically just sums up what I described in the previous slides. The rods and cones are the photoreceptors, and these are what actually starts the process of converting light into a signal. And then the signal transfers from the rods and cones to the bipolar cells, and the bipolar cells then transfer that signal to the ganglion cells. And this is what created the ganglion cells are the first cell to create an action potential. As you can see here, these axons then transfer or come down to the optic disc and then go out, and this is the optic nerve. So these axons actually form the optic nerve. And then in general, there's the sensory input, then there's the process in the retina, and then the response, because the retina processing the information, because the brain can't just take light signals. That's not the brain's language. The brain's language is the difference of sodium and calcium and potassium and all the different neurotransmitters that make up what the brain can actually process. And then that's how we generate an image, and that's what creates vision ultimately. So to conclude, the retina is more than a light detector. It's an information processor. Different photoreceptors serve distinct roles. Rods are for night vision, cones are for sharp color vision. Phototransduction is a unique process that begins neural signaling in an inverted way. Understanding retinal physiology helps explain visual disorders, color blindness, and night vision issues. The brain just doesn't see, it interprets signals already processed by the retina, which is what I mentioned. It's more than just a light detector. The retina interprets a signal first, and then it goes to the brain where it can actually be translated into an image. Thanks for listening.